Good morning, everyone. Thank you so much for joining us today. So glad to be here, and thank you to PCM for inviting me to give this talk. I'm very excited about talking to you about the microbiome and nutrition. I'm also really big into social media, so if you are on Twitter or Instagram, you can @ me at PhD_Leigh, and that's Leigh, and GW has its own hashtag as well. George Washington School of Medicine and Health Sciences, GWSMHS. Okay, how do I use this clicker? The clicker, oh, there we go, okay. Wow, this is a complicated one, sorry about that. Okay, so what is the gut microbiome? The gut microbiome is microbes, and that's bacteria, yeast, viruses, and all sorts of things that we haven't even started to really look into. But what we really focus on in the literature is the microbiome in terms of bacteria. So that's what I'm really going to be talking about today. I'm also really going to be focusing on work that is applicable to humans and applicable to clinical care. There's a lot of emerging evidence that is very interesting, but is not really applicable to clinical care. So I'm going to kind of brush past that. So why do we need to know about nutrition when we talk about the gut microbiome? So the diet is a source of fuel for the microbiota, but it's, how do I go back? Okay. The diet is a source of fuel for the microbiota, but the microbiota is also seeded from the diet. So you're eating food, and hopefully it comes from a nice organic farm, and it's got some great soil microbes on it, and you're eating them, and they're coming into your gut and setting up shop there and doing great healthy things for you. What's really interesting about the role of diet in the microbiome is that some studies have shown that it's almost 5 times more important than the host genetics. So what you eat is almost 5 times more important than the genes that you were given at birth in terms of the microbiome and health outcomes. Now, when we're talking about diet, one of the really interesting things about the microbiome is actually relatively stable. So if you have a short-term diet change, so you typically eat a vegan diet, but you decided, you didn't have any choice and you really needed to eat something at your parents' house maybe, and you had a cheeseburger. Then you go back to your vegan diet, your gut microbiome isn't going to change at all. It will change for that one meal, but then it will come back to its normal baseline. Now, if we do a long-term dietary change, so you became a vegan today, or yesterday when you got here, because we all kind of did. And so now your microbiome has changed, and it's changing with every meal. But if you keep that up, it's going to stay in this new state as the new normal. The number one thing that we've learned about diet and the gut microbiome is that a more diverse diet leads to a more diverse microbiome. You're giving them different kinds of food, and then different kinds of bugs like to eat those different kinds of food. Sort of makes sense. The other thing is a more diverse microbiome is currently the only generally accepted marker of a quote-unquote healthy microbiome. We don't really know what a healthy microbiome is, but if we were going to measure it, we would say it was gut diversity. So thus you can say that a more diverse diet will lead to a healthier microbiome from what we know today. So the microbiome and the human host are in a symbiotic relationship. We eat food, we digest it, but there's a lot left over for the microbiome, and it goes into the large intestine. Approximately 15% of carbohydrates that you eat are making it to the gut microbiome, and that's primarily what we would just consider fiber. That is the fuel for the microbiome. There's a little bit of debate on what else gets there. We know it's very little fat, and there's anywhere from 5 to 34% of protein that reaches to the gut, and probably that has to do with the types of protein, how your body digests it, and how frequently you're eating protein. But it's difficult to separate diet from the elements of diet. The first thing I'm going to go into is calories. So a lot of things we're talking about with obesity is how many calories are you eating. And it's really difficult to separate out the different elements of diet just looking at calories. But when we're just looking at calories, what we do see is if you increase your caloric intake while maintaining the carb, protein, fat proportions, so you're on a very similar diet, you're just eating more of it, what we see is a decrease in diversity. So we know that's bad. And we also see there are changes in who is there. The increase in firmicutes and decrease in Bacteroides, which are generally considered to be markers of negative effects on the microbiome, but this is not as well accepted. So really I would like you to focus on the diversity. So we have a decrease in diversity with an increase in calories. If we see caloric restriction, so you decrease the number of calories you're intaking, but you're on the same diet, we see an increase in diversity. So healthy diet with a low calories, we have a healthy microbiome. But here's where it gets a little confusing. So if we have cases of malnutrition, so say in rural Nepal or in Bangladesh, you're looking at malnourished children, the diversity of their microbiome is actually much lower on a low-calorie diet. So what we think is there's probably a J or a U-shaped curve to this relationship. And as in many things in nutrition, that means there's a sweet spot. You don't want too little and you don't want too much. Okay, so now we're going to dive into the different parts of the diet. So macronutrients, carbohydrates. We're going to talk about fiber and short-chain fatty acids, which from here on I will refer to as SCFAs. I will still call them short-chain fatty acids, though. We're going to talk about FODMAPs, which I'll tell you what that means later if you don't already know. And then we're going to talk about prebiotic supplements. Okay, fiber and short-chain fatty acids. First, I just want to point out that in nutrition, we typically focus on the upper portion of the digestive tract, and we're looking at absorption of macro and micronutrients. But the lower portion of the digestive tract is where the microbiome is, and they're involved in our digestive process as well. They harvest energy from foods that we otherwise wouldn't be able to absorb, and that increases up to 10% of our energy requirements, and that's mostly the digestion of fiber. And when they digest fiber, they produce short-chain fatty acids, such as butyrate, acetate, and propionate, and a couple of other ones. Why do we care about fiber? So fiber, as many of you know, is a bulk-forming element in diet, and it's important for regularity. And that is related to intestinal transit time, how quickly food moves from your mouth through your system. When we have a higher fiber diet, we have a decrease in intestinal transit time. Food moves quicker through your system. And that actually leads to an increased production of short-chain fatty acids. Interestingly enough, increased production of short-chain fatty acids actually increases motility, which then decreases intestinal transit time. So now you have this positive feedback loop to keep you regular. You're eating fiber, your gut microbiome is producing short-chain fatty acids, that's causing a decrease in intestinal transit time. We're generally very happy. One other side effect is there's also a decrease in the pH, which can help with absorption with certain nutrients such as iron. All right, so short-chain fatty acids and obesity. You could see where fiber might have a beneficial effect in obesity. And what we see is a change in the hormone regulation. So we have satiety hormones, hormones that don't make us hungry or do make us hungry. And we see the change in receptors in the gut for them. We see a decreased production in the ones that make you hungry, and we see an increased production in the ones that keep you full longer when you have more short-chain fatty acids. And it also is really important in epigenetic regulation, and this is a really emerging field, so I'm not going to go into detail, but just to show you that it's more than just a pathway thing, it's actually changing the way your genes are expressed. And the hypothesis with this is that the decreased fiber in Western diet could be a large portion of why we see a lot more obesity in our society. Okay, so more about short-chain fatty acids. So I already told you that they're important for intestinal transit time and that leads to a decrease in pH. So we're going to see there's another feedback loop related to the decrease in pH. And that actually leads to an increase in the prevalence of bacteria that produce butyrate, which is one of these healthy short-chain fatty acids. It also leads to a decreased prevalence of pathogenic bacteria, the bad bacteria. And it spares the commensals. So most importantly, your good bacteria are allowed to thrive. And here's another feedback loop that by having those good bacteria, they're producing more short-chain fatty acids and further decreasing the pH and just, again, causing the environment to be very good for good bugs. All right, FODMAPs. What are FODMAPs? So it's a fancy word for a special type of starch that is highly, highly fermentable. The classic example is garlic. If you eat garlic, it's going to get to your gut microbiome and they're going to go crazy. They're going to be so excited and they're going to digest it and they're going to make tons of short-chain fatty acids, but also probably make some gas, and you might have gas and cramp and bloating and pain. And if you have IBS, you might also have diarrhea or constipation. And so one of the temporary treatments for IBS is actually to eliminate FODMAPs from the diet. And I think this is a really great example of what the gut microbiome does and how important short-chain fatty acids are, because this is an example where it may be too much of a good thing. We want to have enough short-chain fatty acids, but we don't want too much. And so if you decrease their intake of these highly fermentable fibers, you're decreasing their short-chain fatty acid production. And you're eliminating their symptoms. But the really interesting thing is the way you detect IBS is very similar to how you detect small intestinal bowel overgrowth, which means we have too much bacteria. So it's a very similar problem, too much of a good thing. And when we take them off the FODMAPs, the microbiome goes back to a more normal state, and we see more short-chain fatty acid producing microbiota, oddly enough, but less production of short-chain fatty acids. So it's like they're normalizing. We don't fully understand how that works, but I still think it's a very powerful example of how important diet is to the gut microbiome and our health. Prebiotic supplements. Basically, prebiotic supplements are fiber. They're all the good things from the fiber put into a pill, and it's really great for your patients because you can tell them to add that to the already healthy diet that they're on, and it will really bolster the probiotic that you're probably also telling them to get. What we've seen, most of the research has been done on two specific types of fiber, inulin and FOS. There's a lot more to fiber than those, but these is just where the research is right now. And what we've seen is that, by definition, prebiotics are probiotic. They are food for the good bacteria. And so we do see that they improve the numbers of good bacteria in your gut. And then we already know that when you improve those bacteria, we're going to improve intestinal transit time. And what is really interesting is we also see that it's anti-inflammatory. We had a bunch of talks about how important it is to have an anti-inflammatory diet. And this may be one of the links between a plant-based diet and anti-inflammation. But here's a really interesting thing. Sometimes it's not always about what you eat. It's about who you have in your gut when you eat it. If you eat resistant starch and you have this one strain of bacteria, you will absorb 100% of the calories from it. If you do not have this bacteria, you will only absorb 20 to 30% of the calories. That's a pretty remarkable difference from just one strain. We don't understand the full interaction between these bacteria with each other, and I'm sure that if you're looking at them in a more complex ecosystem, it's probably not quite this clear-cut, but this was what we saw in subjects that did and did not have this bacteria, who were the same in all other ways. But here's the kicker. It's not even that simple, because every person is different. Your microbiome is almost as unique as you are. It's almost like a fingerprint. And so if we are giving, say, a prebiotic supplement to someone who has a healthy microbiome, they're going to have a very different reaction to someone who doesn't have a healthy microbiome. Or maybe they don't have any reaction at all. And that's kind of frustrating as a clinician. You gave them a prebiotic, you thought it was going to help them, and it didn't do anything to them. And maybe it's because they don't have the right actors in their gut. Okay, so we talked about carbs, and now we're going to talk about fat and protein, and I'm putting them together because largely in the literature they are looked at together as the Western diet. And what do we see in a high-fat, high-protein dietary pattern? It's exactly the opposite of what we see in a high-fiber diet. A decrease in diversity, a decrease in healthy bugs. Oddly enough, there may be a decrease in E. coli. We don't fully understand that. And we see an increase in bugs that thrive off of fat and protein rather than fiber. And that means that we have a decrease in short-chain fatty acids. So we're lowering the food for the colonocytes. But how do you separate high-fat diet from overweight? Because frequently when we're looking at Western diet, these people are also overweight. That's sort of difficult to do. But when we try to tease that out of the literature, what we see mainly with a high-fat diet is actually a decreased intestinal transit time, which is generally good, but the method of that is probably not as good as it would be for short-chain fatty acids. We see a decrease in short-chain fatty acids, but other than that, we really don't know, and it's because it's so difficult to tease out the difference between high-fat diet and high-calorie or obesity. So we've looked a lot in mice, because mice are really easy to control. And this is actually one of the real landmark studies in the microbiome, where they took the microbiome from a subject with obesity and the microbiome from a subject who was lean, who, by the way, they happened to be twins, so they were pretty much identical genetically, and they gave them to mice who did not have a microbiome. And then they put these mice on a high-fat diet, And the mouse who was given the lean microbiome stayed lean, and the mouse who was given the obesogenic microbiome became obese. So we know that that had a lot to do with the microbiome and not the diet, because they were on the same diet. And here's where we think that high fat is maybe less important than the players that are there and the combination of high fat. But it's even more complicated than that. It's the type of fat. As we just heard earlier, it's about the type of fat. So if you take mice and you give them palm oil, you see some bad things in their microbiome. Decrease of diversity, increase of the bad players, decrease of the good players, even some pathogens. But if you do the same high-fat diet with olive oil or safflower oil, you do not see these effects. There's no change in their microbiome. So maybe it's not the high-fat diet. And it's actually possible that healthy fat may, and I emphasize may, be productive in a high-fat diet. So if your diet is high in healthy fats, what we've seen in mice and some studies in children is that your microbiome is not, you don't lose diversity, and the players who we want to see there tend to stick around, and the ones who we don't want to see there don't show up. Okay, so we've covered macronutrients. Now we're going to get into micronutrients. One of the big things when we talk about microbiome is that they actually produce nutrients. So a lot of B vitamins come from our microbiome. And you will see all of these vitamins here, including vitamin K, which is not a B vitamin. The stores in your body are higher than we would predict from the food that you eat, because you are getting a large proportion from the gut microbiome. So just think that over. So you don't even need to eat B vitamins to still get some B vitamins. That's pretty cool. All right, so B1, I don't know how in-depth to go with this with you. I know you guys are getting your intro to nutrition, but I'll leave you with this to study on your own. But thiamine is important for many different things, and one of the main risk factors, interestingly enough, is SIBO, small intestinal bowel overgrowth. So like we were talking about with FODMAPs, related to the microbiome. And what else we know about the microbiome, other than they're synthesizing thiamine and allowing us to have higher stores, is that it contributes to nutritional status. So this is a significant contributor. This is not a small amount. If you remove the good bugs from your body, you will not produce enough thiamine. You will have to take a supplement. B2. We get lots of energy from this, and here we don't see a lot of gut microbiome related risk factors, except for perhaps lactose intolerant, because it's possible they're having some GI inflammation. The interesting thing about riboflavin is that we typically think of dairy as being a source of riboflavin. However, that riboflavin actually doesn't come from the cow. It comes from the bacteria that are fermenting in the milk, or that were on the udders when it was collected. So it's not even that dairy is a source of riboflavin, it's that bacteria are a good source of riboflavin. And we believe, it appears that your gut microbiome is a significant source of B2, but we're still figuring that one out. B6, not something we typically see in terms of nutritional deficiency, but it's really important for your microbiome. They actually depend on us for their B6. They need it for enzymatic activities, which means it's a pretty systemic effect. Unfortunately, there has not been a lot of studies on this, probably because we don't see a lot of B6 deficiency in our patients. But what is really interesting is there was a study showing that there's a potential relationship between how virulent and how easy H. pylori moves around and its ability to make B6. So if it can provide its own B6, it's more virulent and more motile, which is potentially dangerous for our patients. So an interesting food for thought. I don't know how applicable it is yet. Folate, very important. Here again we see SIBO. So you're going to see that in a few places. If you have small intestinal bowel overgrowth, you're going to have a risk of some B vitamin deficiencies. We believe that the amount produced by the microbiome is significant, but it's not totally clear on that yet. We do know that resistant starch is really important in the production of B9, folate. So if we don't get enough resistant starch in the diet, you're probably not producing significant amounts of folate in your gut microbiome. That may be an area of supplementation for your patients. However, there's a fairly large caveat for that. One study has shown that patients who had increased production of folate in their gut had increased concentrations of folate in their stool, meaning they probably didn't absorb most of it. So there's a little bit of a caveat as to whether supplementing with resistant starch may improve folate status, but resistant starch is good for you and it's probably not going to hurt, so why not? B12, it's a big one for this group. Probably need to be supplemented with that one. And here again, look, we see SIBO. So the gut microbiome is important in this as well because the gut microbiome produces B12. But it's also crucial for the metabolism of the microbiome itself. They rely on us for B12. And it has something to do with our epigenetics and may be the way that the host and the microbiome interact in an genomic fashion. But what's really interesting is that in one study, so this is just one study, 83% of the gut microbiota were shown to require B12. It was essential for them. So if you aren't providing enough B12 for your gut microbiome, these bugs are going to die. So you need to make sure that you're supplementing if you aren't getting enough from the diet. And we don't know if the requirements of the gut microbiome may contribute to B12 deficiency, but it's possible. K1, or vitamin K. So we always say vitamin K, but it's actually vitamins K. There are two different main types. What we traditionally know is vitamin K1 from our green leafies, which everyone in here is getting enough of, so we don't need to worry about that. But K2 is the form that's becoming more popular in the literature of the microbiome because it's actually produced by the microbiome. So green leafies have lots of vitamin K1, which you can see with that large blue bar, but things like natto, which are fermented, have just massive amounts of K2, which you can see from that big green bar. So we maybe don't need to eat a ton of natto, but you should probably get some of it in your diet to get your K2. And this is like the storage form of vitamin K. And we've already started to see associations with heart disease and osteoporosis, and there are many other that are emerging, so making sure you get enough K2 is very important. Like I said, it's produced by the gut microbiota, but they also require it. They actually use it as an electron carrier. So again, if you're not getting enough K2, your bugs are not getting something they need to live, and they will probably not be healthy. Vitamin A. So I would be remiss as a graduate of the Johns Hopkins School of Public Health if I didn't mention vitamin A and eye health. We discovered that role, and it's very important. It's unfortunately maybe not super relevant for you living in the United States where we don't see a lot of vitamin A deficiency, but it is really important in developing countries where there's a significant burden of disease with vitamin A deficiency. And I think scientifically it's very interesting to see what's coming out of the literature. We've found that the gut microbiome can produce beta-carotene, which is great, but we all know we need retinol. We need the active form. And we haven't shown that bacterial beta-carotene is activated into retinol yet. So we don't know if they contribute to vitamin A status. But a really interesting bit of trivia for you. If you haven't heard of it yet, B. infantis is going to be all the rage for OBGYN in the future. And it is the bug that we have found that is largely missing in modern Western infants. And when you supplement with it, it can cure what ails you, pretty much anything. It's highly anti-inflammatory, it prevents autoimmune disease, asthma, atopic dermatitis. So it's going to be very hot very soon. If you haven't heard about it, go out and look about it. But the effect of B. infantis is dependent on having sufficient vitamin A. So if you deliver B. infantis in an infant that is vitamin A deficient, it doesn't do anything. So here's why vitamin A may be important. If you have an infant who may have some sort of risk factors for vitamin A deficiency and you're trying to treat them for some sort of inflammatory condition with, for instance, B. infantis, you need to make sure they have sufficient stores of vitamin A. But if they're getting enough orange vegetables, they're probably okay. Vitamin D, I'll tell you this is the nutrient that's closest to my heart. It is maybe not super relevant for the microbiome because they actually don't have a vitamin D receptor, but vitamin D is crucial for your immune system. And the way your immune system reacts to commensal bacteria, the good guys, or reacts to pathogens is dependent on vitamin D. So if you don't have enough vitamin D, you will have an inflammatory state or your body will not fight off the infection well enough. These are not good. So go outside is my number one recommendation, but if you don't have time to go outside because you're at an awesome conference like this, take your vitamin D supplement and it'll be okay. Okay, we're getting down on the micronutrients, I promise. Iron is largely not well explored. What we know is that iron deficient patients seem to be lacking lactobacilli. We don't understand the directionality of that relationship, but we do know that lactobacilli requires iron. So it would make sense that a patient that was deficient in iron wouldn't be supplying enough iron to their gut bugs to be happy. However, there hasn't been any supplementation studies showing that something with iron would improve that. But we do know that having a healthy microbiome produces short-chain fatty acids, which decreases pH, which increases absorption of iron. So there's a directionality issue. But maybe if we switch them to a whole foods plant-based diet and got them taking a probiotic and maybe even a prebiotic to really get them revving, that they'll start absorbing iron better, and that will fix some of the issues of not having the right bugs. Zinc. Basically all we know is that there's been one study of supplementation with zinc that showed an increase in what we generally consider to be good bugs, firmicutes, particularly lactobacillus. But that's really all we know. Zinc deficiency is fairly rare, and when it occurs it's very mild, so I'm not sure how clinically relevant this will be, but something to be aware of, that there is a connection. Okay, so we're done with the microbiome and micronutrients. Now we're going to move into the other components of food, which is something I think is going to be huge in the microbiome. I typically refer to them as non-nutritive bioactives. Many of these will sound familiar to you. Polyphenols. We all know what a polyphenol is. That's why we want a whole foods plant-based diet. And these have been linked to many health benefits, which you've already heard many times today. But the really interesting thing is these polyphenols are largely not absorbed by you. They make it to the large intestine. 90% of them make it to the large intestine, and they're likely bioactivated by the microbiome. So a lot of the beneficial effects of the polyphenols may actually be due to their effects on and through the microbiome. All right, wine. We all love wine. It has lots of great polyphenols in it. Some ones that you're probably familiar with is quercetin, which is also found in apples, caffeic acid, which is also in tea, and resveratrol. That's the big one that we talk about when we talk about wine. But it has many different polyphenols. Foods are really complex. So when we try to figure out what's going on, we try to pull different elements out and do a more reductionist scientific approach. This is a very great research study where they tried to pull out the effect of the polyphenols in red wine and the effect of alcohol, ethanol. And they did that by delivering wine, dealcoholized wine, so basically all the polyphenols without the alcohol, and then gin, which is just pure alcohol. And what they saw was an increase in all the good bugs, but what was most really fabulous about the study is that the really significant increase was in the wine as a whole food, and it was synergistic. So the effect of the wine and the effect of the alcohol added together was not equal to the effect of whole wine. Putting them together got a much larger effect. So having a glass of wine versus a glass of grape juice is not going to be the same thing. And if we try to isolate wine further down to single polyphenols, there is a few studies, and we're seeing a lot of beneficial effects, improvement in commensals, decrease in pathogens, but I would argue that this is sort of missing the point, because people eat foods, not compounds, and there is this synergism in foods called the food matrix, right? So when you're having a glass of wine, you aren't drinking flavin-3-alls, right? I don't know how relevant it is. I understand as a scientist we have to do this, and it's very meaningful, but I'm not sure. I feel like it's kind of missing the point a little bit. So we're going to talk about some more whole foods. Now, obviously this is complicated to study because it's a whole food. We don't know what part of it is causing the effect. But berries, we all ate great berries this morning, and you will be happy to know that your microbiome is happy about that. We know specifically that some studies have shown that a few bacteria that we consider generally to be good increase, and the ones we generally consider to be bad decrease. Most importantly, there are some studies showing that berries may actually weaken salmonella by increasing their membrane permeability. So that's an actual mechanism of action we know that berries have on these pathogens. Not well explored, but very promising. Tea. So tea has a whole bunch of great polyphenols, and I'm very pro tea, which you probably know if you listen to the Exam Room podcast, because I already talked about green tea on there. And you're seeing very similar effects. It's limiting pathogens. It's sparing commensals. What's really interesting about this study is that they showed an effect on C. diff, which I don't know about you, is the one that scares me the most. So if there's something you can do to prevent C. diff, overgrowth in your gut, I'm going to do it. And I'm going to keep having my green tea every morning because this is great motivation. However, this has not been well explored. So I wouldn't say this is gospel at this point, but it is very promising. Cocoa, actually a personal favorite source of polyphenols. It has many, many, many kinds of polyphenols and tons of them. So if you're eating your tiny square of dark chocolate every day, like I do, you're doing good. You're limiting the pathogens, you're increasing commensals, but this is actually even less well explored than some of the other areas. And I think part of it is because not only is chocolate a complicated compound in having so many different polyphenols, but what kind of chocolate, right? How dark does it need to be? Does it need to be 88% or is 77% enough? Or is 72% okay? So I feel like that's a major barrier to studying chocolate, but we have a lot of motivation for studying chocolate, so hopefully that will overcome it. But so what's the take-home message? That unfortunately controlling diet in humans is difficult, and doing research studies where you're controlling diet is even more difficult. And so that's why some of this research is quite limited. Much of the work has actually been in animal models for this reason, because you can tell an animal what to eat. However, it has extremely poor translation to humans. Animal studies frequently don't translate to humans in general, but especially in the microbiome, it does not seem to translate. It's just a different environment. Some of the best work we have is done in small feeding studies. They're highly controlled. We know exactly what they're eating. It's measured. It's designed, as the muffin study was earlier, designed for the question. However, that means there's a very small sample size. And with a small sample size means there's a very limited variety of subjects. And as I told you earlier, your microbiome is unique. So if we have a very small variety of subjects, we're having a very small variety of microbiomes. And so is this really generalizable to the general public? Hard to say. Finally, supplementation studies. These are going to be some of our easiest to answer questions, our most well-controlled. However, as I said earlier, we eat foods, not compounds. So I don't know how relevant they are, and they're definitely not taking into account the food matrix. So if we could do some sort of supplementation trial with a whole food, that would be really fantastic. All right, so what do we know? What don't we know? Basically, we don't know a lot. We focus mostly on fiber and short-chain fatty acids, and there's a lot more to nutrition in the gut microbiome than fiber and short-chain fatty acids, though those are very important. Micronutrients, or macronutrients, we haven't explored the role of fat very well. We haven't explored the role of protein separate from fat. And then what about non-fibrous carbs? What does sugar do to the gut microbiome? Something we needed to figure out. Macronutrients, there's a lot to be learned, and just the ones that I listed here are ones we have some emerging evidence, but there's likely other micronutrients that are important. Polyphenols, I alluded to how we've started to explore this, but there's so many polyphenols out there, so many phytonutrients in the many different plants that we eat, and many of them are likely beneficial. And here's one that's also kind of close to my heart, food additives, so processed foods, things we're adding to our food that haven't been in our food system. Maybe they're okay, maybe they're not. Something I personally worry about is someone who's gluten-free, because a lot of the gluten-free substitutes have things like guar gum, which I don't know what effect that has on my gut microbiome. Maybe it's okay, maybe it's not. Something we need to figure out. And then contaminants, pesticides, carcinogens, heavy metals, not good for us, probably not good for the microbiome, but that has not been studied. So what do we need to do? We need human trials. We need to find the relationship in humans, not in mice. And then once we've found the relationships in humans, then we can look at mechanism. We can design studies to effectively look at mechanism because we understand the relationship in humans. All right, many thanks. This is part of an upcoming systematic review that I'm publishing with a grad student from GW, Elise Costa, and Scott Jackson from NIST, who, full disclosure, is my fiance. We fell in love over the microbiome. And also GW Clinical Research and Leadership, which is my department, and they're wonderful. I have a great department chair who's highly supportive of not just integrative medicine, but research on whole foods, and I think she lets me think outside of the box, which is wonderful. I also want to thank Janet Rodriguez, who's my partner in crime for the Office of Integrative Medicine. I didn't put you on the slide. And a plug for our podcast, if you haven't seen it out there. It's actually the GW Integrative Medicine Podcast, so if you're trying to search for it, GW Integrative Medicine Podcast, not GW School of Medicine and Health Sciences. It comes out once a month. We do really cool things, like I did one on nutrition and the gut microbiome. We've done things about yoga therapy. We talk about acupuncture, medical cannabis, anything that's like hot and people don't really know the answer to. We try to talk about it.